cycles from theory-backed X-ray spectroscopy. I am a spectroscopist by training, and I have the role at SSRL of really driving DOE BER science, and I'll talk a little bit about that through my presentation. This is the outline of my talk. So I'm going to first tell you about how SLAC SMB, which is a structural molecular biology group, supports DOE BER mission science. I'll give you a snapshot of our facilities and capabilities. I'll then talk about our methodology of supporting BER researchers through an innovative outreach program that we run there. And then I will talk about some really neat results that have come out of our collaboration with EMSL theoreticians. And I'll end with some future work. Okay, so I'll start with the acknowledgement. We've several people from SSRL and EMSL who are involved in this work. We have scientists who are actually involved in the science, and then we have SSRL spectroscopy group who are, behind the scene beamline scientists who enable this work. So I particularly would like to call out Sharon Bone, Sherry Yu, Joss, and Macon, who are involved in the science. And then at EMSL, we have worked with Amity Anderson, Hoshin Kim, and Boyana Janovska here. And we've got a lot of help from Satish and Jay and Neri in kickstarting this collaborative work. We've also worked with Joss Cass at University of Washington in bringing some of these theory tools to our collaboration. Okay, so I want to give a broad overview of DOE BER mission-inspired science. To the left here, you can see some scientific thrusts of the Biological System Science Division within BER. And just looking at these scientific thrusts, we can see the ambitious goals of BER in trying to understand and replicate biological systems for bioenergy production, but also moving beyond that and trying to engineer these systems and modify them for new biotechnology breakthroughs. And as scientists, we understand that this can only be achieved if you understand the biological, biogeochemical, and physical principles that underlie these processes. And what you really need to do is have understanding across the spatiotemporal resolution. You want to understand genome, molecular scale, understanding of systems, all the way to earth and environmental scale systems. And so here, while BER's mission is this wide, I wanted to highlight that synchrotron techniques, especially the ones that we have at SSRL within the structural molecular biology group, sort of focus in this purple box. So we are really good at looking at processes starting from sub-nanometer range through centimeter range on tissues. And with addition of the free electron laser, we can really look at femtosecond through days processes. And here to the right, I'm showing some scientific thrusts that can be tackled with our techniques. We can look at how metalloenzymes work. We can look at how microbial communities are responding to stimuli. We can trace heavy elements through their biogeochemical processes. We can look at nutrient uptake by roots, and also look at biomass degradation. So it's a wide variety of science that synchrotron sources can address. And in particular, the structural molecular biology group at SSRL has addressed in the past for our users. Okay, so this is a broad overview of the tools that we have at SLAC. Okay, this was an animation, which of course is in a PDF, does not show up. So you just have to imagine as I tell you what this is. So in the center, I have these bubbles, which are really representing the structural molecular biology group within SLAC, which is the home of synchrotron science within SLAC, which is, a national lab. We have 4 different scientific techniques that we make available to our users: macromolecular crystallography, small angle scattering, X-ray spectroscopy and XRF imaging. And so with that, that's been a stronghold at SLAC in terms of supporting BER users. We also have two newer facilities. One is one of the NIH cryo-EM centers. So we have S2C2 for collaborative imaging work, and we also have LCLS where you can do serial crystallography. So together, all of these, through all of these, we provide sort of a holistic approach to looking at biological systems at various timescales. And so if I clicked this right, you would then see all of these images, which are really showing that with our techniques at SLAC, we can address a variety of sample size and complexity. So if I look at this very right-hand side rhizosphere system, which is more complex and larger, we can look at, as an example, metal uptake by roots and their transformation as they're taken up and delivered to various parts of the plant. But then on the other side, we can drill in and look at the electronic structures of the metalloenzymes, which are responsible for many of these plant-based catalysis. And in between, we have several other techniques which really nicely map the structure and dynamics at important resolution levels, such as crystallography, SACs, and cryo-EM I told you about. And as a facility, we are funded by various different sources, BER, NIH, and BES. And the reason for me to mention this thing is because all of us are doing science, which are the interface of all of these various funding agencies and their missions. And so we sort of span the gamut through our resources. Okay, so a little bit more detail about the 4 techniques that I mentioned. As I said, we have macromolecular crystallography. We have 2 leading beam lines, 12.2 and 12.1, which have intense microbeams for very small crystals. We have full automations and robotics and remote access, so you can remotely send in samples and remotely measure. The new capabilities that we have now are, physiological temperature measurements. You don't have to freeze your crystals. And we are pushing multimodal in a big way. We have in situ optical and X-ray spectroscopy methods on our crystallography facilities. We have small angle X-ray scattering, which essentially provides 3D structures of biomolecules in solution at slightly lower resolution, between 5 to 10 angstroms. This is really great at looking at conformational changes and dynamics in solution of macromolecules. And here too, we have a lot of automation for sample delivery and high throughput measurements. Then we have a combination of X-ray spectroscopy techniques and X-ray fluorescence imaging techniques. What really makes SSRL unique with both X-ray spectroscopy and imaging is that we have dedicated instrumentation for biological science in the tender X-ray region, so we can probe from phosphorus onward all the way to 30-35 kV. So we can cover a variety of elements, starting from, we heard about NPK a lot here. We can really look at phosphorus, we can really look at potassium, and look at how they're changing in various systems. And the other thing with X-ray spectroscopies is that we have a combination of quantum chemical tools that we make available to our users. We understand that not everybody is an expert in theoretical tools, and we either support them through the SMB facility or through collaboration with other experts in theory. And I'm going to talk about 2 of those examples here. Just to dwell on the X-ray spectroscopy a little bit, SSRL has 8 X-ray spectroscopy facilities, which are all under the same management, which means that we have the same software, same protocols across these beam lines, and we can move our biologically relevant work to any of these beam lines if the user requests that. So that's a big flexibility that we provide to our. Okay. Doing research at SLAC SMB facilities, we have a query to results pipeline, which frankly most synchrotrons have a pathway where you go in, you have a scientific query, you become a user, write a proposal, and then you measure data, analyze results all the way to reporting. And most places have really nailed the measured data. Most of the synchrotron facilities that you have across the US and in the world are really working hard to make beamlines that will get you really high quality data. Fun. Yeah. Okay, so but this is often not good enough for new and novice users. And so we have a different pipeline for new and novice users, especially for DOE BER research, because we were funded to do that by BER, where we really help understand our users, the strengths and limitations of our facilities, and really work with them through their measurements, and sometimes find collaborators for them. More recently, in the last 3-year period, we have a dedicated, as I said, a dedicated outreach program, which is again a DOE BER funded opportunity where you as a user can come in, and we have a variety of staff members who have different skill set, who are going to walk you through developing the scientific case all the way through manuscript preparation. We can do that collaboratively, where our scientists work with you and our partners in the research, or we could work with you in really just supporting you through the process. And the goal there is to transform the novice researchers to becoming an independent user in a few proposal cycles. And this has really taken off. We have really grown our BER footprint through this dedicated initiative, and it's now being successfully replicated at other national labs. And we are really excited about bringing our tools to all of you. Okay, so as I said, I am a spectroscopist by training, and while our BER initiative was happening, where we were finding all these users, we really started working with theorists here at EMSL to figure out how to explore an SSRL-EMSL collaboration. So we were working at that time with Nancy Hess, who was no longer here, and Niri Govind, to really figure out scientific areas where we could really bring EMSL and SSRL technologies to our user base. And one thing that was clear to us was that even though we are a BSSD funded facility, we were putting out a lot of science in what we call this interfacial region between BSSD and ESSD in the environmental microbiome, sustainable biogeochemistry, and biogeochemical cycles. In the last sort of 10 to 15-year period, we have upward of 500 publications from users who are coming to SSRL and doing science that is broadly related to this area. And so this was sort of an opportunity moment for us. And we said, all right, we're going to figure out a collaboration between SSRL and EMSL. EMSL has great theoretical tools. You have this supercomputing facility. And so what we said that we could do spectroscopy at SSRL, and we could do modeling at EMSL, and we ran several workshops which were very well attended, and really figure out the needs of the user community in collaboration with various folks here and at SSRL. And so it has really taken off in the last few years. We have several publications that have come out in a collaborative manner. between SSRL and EMSL folks. And this is sort of a nice schematic that Joss made for us to really highlight how biological systems are everywhere in the interface of environmental and biology, and we started looking at systems through this collaboration. Okay, so I'd now like to talk about the two science examples that I have prepared here. But for those of you who know X-ray spectroscopy or are new to it, it's essentially an element-specific technique which can be applied to heterogeneous systems, and you get two types of information. One is XAFS, which is probably the more familiar method, which essentially tells you you're zooming into a metal site, is going to tell you how many of what type of atoms are at what distance from that metal center. So it's going to give you atomistic level structure details around a metal center or an element center. It doesn't have to be a metal. And then you have a lower energy Zanes region, which holds electronic correlation about your system. If you can analyze it really well, you can actually understand that these transitions all occur due to, the bonding that's happening in the molecular system and transitions from metal 1s levels to the various bonding levels. And their intensity and energy distribution tells you about what that system is. And typically you put these two together, you bring density functional theory or other methods to try to obtain a holistic picture about the system that you're studying. And here I have a metalloenzyme picture showing that we are applying these techniques to metalloenzymes where you have discrete molecular systems. Our goal is to bring that to the more messier biogeochemical system, right? Like the systems are heterogeneous and you can't really say there's only one species, right? And so how can we apply these methods to these messier systems and get beyond pretty pictures of elemental distribution? So this is an elemental map of, I forget now what it's a plant system where we are looking at the total iron distribution. But we want to go beyond that. We want to zoom into, in these various spots, look at the spectral shape, and then say, okay, one of them is this and the other is this. And so we want speciation map, not just elemental map. And we want to bring theory to help with that. So as I said, I'm going to talk about two projects. The first is a combined spectroscopic and theoretical analysis of organic potassium compounds. The motivation of for this work really came from a collaboration that one of our group members, Joss, started with Arunima. These two are doing fantastic work on this terraforms, which you'll also visit tomorrow, and this is one of their early work where they were growing these fungal hyphae on these terraforms and looking at how mineral-derived micronutrients are transported across these hyphae. So this is one of their terraforms, and you can see the fungal hyphae sort of growing around these posts. And when you look at the mapping, you can see there's a lot of organic potassium. Did I say phosphate? This is organic potassium that is around these posts, showing that that the fungal hyphae is converting mineral into organic species, and the mineral potassium is distributed across in the micromodel as it was made. And so here I'm showing you the spectrum of organic potassium and potassium citrate, and you're seeing them they're quite distinctly different, and that's what allows for this differential mapping. When I first saw these spectrum, it was quite a disbelief for me because all known potassium compounds have 6 to 8 potassium oxygen bonds, which are often sort of disordered, 2.6 to 2.9 angstroms. And if you understand X-ray spectroscopy, you would say all the spectra should be more or less the same. for potassium. They should all kind of look similar, maybe some shifts, some intensity changes, but I was not ready for this, really big change in spectral shape in this unknown hyphae that we looked at. So the question was, why are the Zane spectra so different in these species? And here I'm showing you, a variety of potassium that's around in, minerals and biological systems. They're all, 6 to 8 coordinates. But it gets better. We measured a bunch of potassium species tartarate through an unknown humic acid. So we did tartarate, oxalate, citrate, nitrate, acetate, and you can see that the Zanes are very rich with 6 to 10 distinct features across the rising edge, across this 15 EV region. And what we were saying that while these are structurally similar, again, structurally, there's 6 to 8 oxygens around that potassium, are these X-ray spectra telling us that they are signatures of the chemical form of the species? Like if I looked at this, could I say, okay, that's citrate and that's nitrate because there's something interesting going on in their bonding? And our hypothesis is that we could use those, we could step back. We could use DFT or theoretical methods to really reproduce these spectral changes. None of these tools had been applied to potassium spectroscopy. And again, I'm choosing potassium and calcium as an example for this audience because SSRL is really good in that energy region, in the tender XAS energy. And while we've also applied this to iron and manganese and other systems. And we are also, one of our hypotheses here is that perhaps doing this analysis, trying to understand where these transitions are coming from, is going to ultimately help us bracket the potential species that contribute to unknowns or humic acids. And I'll tell a little bit more about that. Here we collaborated with EMSL. We worked with Amity Anderson here at EMSL, and then we worked with, Joss Cass at University of Washington, and we applied 2 different methods. We applied DFT-based methods, and we applied multiple scattering-based methods that are part of the FEF10 code. And I have 2 figures that we prepared for our publication, and I can go into very much details about the density of states that are shown in this, but in the interest of time, I'll focus on 2 things. The first is that full multiple scattering based methods are excellent in reproducing Zanes, especially for systems that are mineral like. So for example, here you can see that the entire spectrum of potassium nitrate has been well simulated with all the features reasonably well shown. While DFT-based methods are not really good in the very high, in this 15 EV region. DFT-based methods are really good for lower energy transitions, and I'm going to talk about that in the calcium example. And it turns out that the DFT-based methods are become more accurate for molecular system or metal ionic interactions, and we're looking at potassium in solution to really compare these two. But more importantly, what these methods allow us to do is assign spectral features to bonding. So I'm again plotted the same comparison spectrum of the various species and pointing out nitrate and citrate. You can see that there's a very low energy feature in potassium nitrate, and the density of states in our calculation shows that this is very much associated with this bite angle of the nitrate and on potassium, which gives shifts this feature to lower energy. And so here we are saying that these low energy features occur because of very strict structural criteria, and if you see such features in your unknowns, you will significantly narrow the candidate pool that contribute to that feature. Similarly, in our citrate calculations, what we were able to show that this feature that appears in several of these compounds is because of water coordination. So in citrate, in potassium citrate, there's a water molecule. And in our simulations, if you include it or you don't include it, this peak changes. And so we have a hydration indicator in the potassium spectrum, right? So even if we are not necessarily able to tell you exactly which all species are there in the humic acid or unknown species, and we could do all amounts of linear combination or principal component analysis, but if you don't have a good basis for the species, then applying DFT, which brackets, okay, these are the species that could be, is really helpful. Okay, so I'll now move to the second example on calcium containing materials. So the impetus for this work was really brought to us by Sharon Bone, who's working on calcium speciation in environmental systems. And her question was, how do we assess chemical forms of calcium in complex heterogeneous systems like soil? She was using microspectroscopy at SSRL. So you take a soil sample, you get a spectral map on one of our imaging beamlines, you'll identify where calcium, sulfur, and phosphorus are located. You would zoom in, and then you would get the spectrum, which we could call calcium organic matter XAS, or general calcium unknown XAS, because you don't know what's going on here. And then we compare it to various reference compounds. And in this plot, you can see these reference compounds that we have chosen are mineral-based compounds. You can see they're very spectral rich, and the data from the soils are sort of broad. And that was the reason for calling these organic matter. They have to be different from these reference mineral compounds. And ultimately, there's some sort of linear combination analysis or fitting to narrow this speciation, and sort of do this in this business. And so we were saying, okay, this is clearly not a mineral spectrum, maybe a mixture, but there's other things that are going on there. But another thing that's really interesting is if you zoom into this pre-edge region, which is right at this 40 EV, you can see that there's a shift in energy, and there's this double peak here. And we were interested in understanding, can we use that for to understand more about what system it is. And we wanted to start with something that was very fluffy. We wanted to start with solvated calcium as our model system. And I think I'll go to this and then come back to that, where solvated calcium does not have any sort of long distance effect from a mineral. And otherwise too, solvated calcium is very important because calcium is relevant in biological processes, environmental processes, and in the rhizosphere. And one of the ways calcium is biomineralized or is uptaken into the rhizosphere is starting as calcium in solution, however transiently it's likely bound to water molecules, and then transfer to a protein molecule or biomineralize, or so on and so forth. So I'll go back to this. So when we started looking at just solvated calcium, right? You would think that that is a question that's been reasonably solved, and that is right. There's a long history of literature on the structure of solvated calcium molecules. However, there isn't much of a consensus. Theoretical tools have been used which predict calcium oxygen coordination numbers of 6 to 10 and bond distances between 2.32 to 2.5. So a wide spread of how many waters are around this calcium. XAPS data are available, which shows coordination numbers varying from 7 to 9 over a tighter bond distance spread. However, calcium K edges have a 2-electron transition in their spectrum, which really messes up with their XAPS data. So XAPS modeling is a little bit hard because of this 2-electron transition that's right smack in the middle of the XAPS spectrum. Furthermore, XABS has a about 25% bond distance uncertainty, especially when you're measuring at room temperature and haven't cooled down your samples. This 7 to 9 is really 6 to 10, right? So it wasn't really known whether how many waters are around calcium, right? So our hypothesis here was that theory-based methods can differentiate between and can predict the bond distance and spread, and ultimately we could use this study to create a framework for interpretation of more complex things. Okay, so looked at that. So we wanted to approach our modeling of our calcium, solvated calcium, in two ways. We first wanted to do an inner shell structure where we wanted to use DFT to do optimizations of 4 to 10 waters in the solvation shell of calcium. Created these models. And here we were saying, well, if we calculate these structures, we'll get a bond distance spread, but we will also get a spectrum that we can calculate that will uniquely tell us about the ligand field splittings happening in the DD levels at calcium. And that will help us correlate with the experiment and really tell us which of these it really is. And, we went down to 4. Nobody's saying calcium is 4 coordinate, but, that's the advantage of DFT. You can put in whatever. Okay. So from our DFT results, we looked at the structure, and our optimized structures really show that if you put from 4 to 8 waters, you'll sort of optimize to a 4 coordinate to 8 coordinate. But if you start putting in more waters, 9, 10, they sort of fly away, and you go to 7 coordinate or 6 coordinate. Essentially, what the DFT is saying that it agrees with at most 8 water molecules around calcium. Then we looked at the TD-DFT correlation of that tiny pre-edge region, which I showed earlier. And what we noticed that if you have 4 to 5 waters, the intensity of this feature is really too high. You can see a mismatch between experiment and theory. That's very normal. DFT methods cannot predict the core potential really well. Okay, so you can see the 4 and 5 coordinates are very highly intense. And so without doing any XAFs, you already know that it's not 4 or 5 coordinates if you had this DFT. It can also not be 6 or 8 waters because they have 0 intensity. So 8 water and 6 water, they have 0 intensity. It can only be 7 or 9 waters, and 9 water actually optimizes to a 7 water model. So essentially, again, without having any structural probe, you can do Zanes and DFT to say that calcium is actually has 7 waters associated with it. The bond distances that we get from a DFT really match all the experimental data that are out there. I point out here that our intensity is a reasonable match, but is slightly lower. And so that's where we started looking at outer shell structure, where we did AIMD optimization of calcium with 40 waters, so with 7, and then we increased that to 40 different waters. So lo and behold, what turns out is the best structure has actually 7 waters in the first shell, and that is spread across, this 2.41 to 2.46 angstroms, which is consistent with experiment. So there's 2 observations here. The first is that this is what we looked at, the pre-edge earlier, and when you add 40 waters, the match actually becomes quite good. Essentially, the outer shell is impacting the pre-edge region also, which was a surprise because this is a purely electronic feature. But in addition, you can see that the addition of more waters really helps in predicting the rising edge, which is not at all predicted with only the first shell. So then what we did was we said, okay, we looked at the 40 atom model. Let's do a second solvation cell. So we started with 7. We added a few more up to 18 waters. And we see that the second shell does a reasonable job at reproducing the pre-edge. And it turns out that there is enough electron density that's spread across into the small electron density spread into the second shell, which actually contributes to some transitions in this region, and that's why the simulation is better. But also, our calculations really show that the larger model is required to predict the rising edge because that has, long range multiple scattering. Okay, so this is sort of, again, summarizing what I just said. 7 water model. This is the tiny pre-edge. Down there, you can't really see, but this doesn't quite look like an XAS spectrum. When you start going to the 40 water model, it starts looking like an XAS spectrum. The other thing that we noticed, which was also a bit of a surprise, is that multiple transitions contribute both to the rising edge and to the pre-edge region, but really the diffuse density contribution from further out waters gives this intensity into the pre-edge. And in particular, this is important for weak ligands such as water molecules when they. So we are looking at the fluffiest of system, waters around calcium. It will only get better when we look at molecular systems, which is our next thing. So we want to now apply that to a variety of calcium compounds, phosphate, sulfate, carbonate, etc., and try to predict the differences here that we see in the pre-edge, and then look at the zanes too. So we're taking 2 approaches to expand this to organic matter. One is, really apply the technique that we have to all of these potential systems that are present in organic matter, or we take sort of a blind dive where we assume some of these structures using AIMD methods and then model that through comparisons with mass spec data that are collected here at EMSL. Okay, so that's where we are at with these stories. And the future outlook, we were very glad that one of our collaborations was awarded the large scale research project here this year. In this project, we are working with Roland in Michigan State, and we really want to bring again this collaborative work where we do copper edge and XFs on at SSRL. We do large scale modeling at EMSL. We also have proposed some NMR to be done at EMSL to really try to understand copper binding to this very unique enzymatic system. So I showed you that the potassium spectra where, look how cool, I'm still like amazed by how coolly different they all are. And one of the future goals is to write another LSR here. And that you'll hear more about that from Josh, who's presenting, a couple talks from now. Please attend that, and she'll talk more about the reason behind doing all of these theoretical works. So with that, thank you for your attention, and I'll take questions.
Riti, we have time for one question.
Thanks so much, Riti. That was really great. Let's see. So for folks who are interested to get involved and start doing measurements, they haven't used synchrotron before to measure biological materials. You showed this pipeline to getting people started within a few proposal cycles. Who should they contact, and what's kind of the process if they're just developing an idea?
Right. So they should write to us at beroutreach@slac.stanford.edu. If you are not funded by BER, don't worry about that. We're just, please write to us. So you would describe in an email what you want to do. It may be one of the 4 techniques, and we have staff members who we can assign to you, who will then work with you to figure out, one, if this experiment even makes sense at SSRL. If it doesn't, we take an expansive view. If it doesn't work at SSRL, we might, connect you with folks at ORNL or ALS or APS who we know there's some technologies that are available there. But let's say we figure out that, yes, this will work here at SSRL. That is where earlier we used to stop. Used to say, yes, it'll work, apply to this beamline, right? But what our scientists then will do is sit with you and develop the proposal that you write, and then work with you through that rating system, and at the end, analysis. The important thing there is that the users, the new folks who come in, have personnel who are willing to drive that on your end too. Like it's collaborative. So hopefully students are willing to learn as we are setting them up to become experienced users.
How about feasibility data collection prior to the proposal?
So we do have several mechanisms. We have a rapid access mechanism where we do feasibility tests. They had sort of gone away during the COVID period, but we are bringing them back in. We have 1 day of beam time, which is plenty for feasibility tests. We will have that mechanism for new users. Thank you.